Welcome to the first of the scientific sessions. We just finished our diversity session, which was really interesting and really fascinating. Now we're going to do 3 talks, all on the broad theme of learning and memory. The first talk is going to be Christos Constantinidis from Vanderbilt University. Christos did his undergraduate in Greece and then came to Johns Hopkins for his PhD. Then he moved to Yale, where he was part of this really amazing generation of monkey physiologists that have kind of really revolutionized the field. People who came out of Pat Goldman-Rakesh's lab. I've been following his work for a long time. I got to say, his papers are just really amazing. I'm also a monkey physiologist. Every time you read one of his papers, you kind of learn something, even just beyond what's in the title of the paper. There's just a lot of creativity there. But recently, he's also started doing human stuff, and so we're going to learn a little bit about that blending of ideas and how we can take monkey ideas and improve humans. So welcome.
Hello. Thank you very much, Ben, for this wonderful introduction. I'm not sure if I can live up to the expectations now after that. And I want to thank the organizers for inviting me. It was wonderful to meet so many friends and colleagues here and be exposed to the refreshing Minnesota weather. It makes you feel alive again. So, as Ben mentioned, my background is in neurophysiology using non-human primate models. And today I want to talk to you about a series of experiments that we've done the last few years, relying on the non-human primate as a model to develop deep brain stimulation, targeting the cholinergic system as a way of improving cognitive functions with an eye into translational applications and eventually moving that into the clinic, in the human applications of assisting with impairments of memory and cognition. So in the first part of my talk, I'm going to talk about the mechanisms of action to some extent. So in order to understand the action of this deep brain stimulation we use, it's really essential to understand the mechanisms of cognitive functions and working memory. And once we have at least some mechanistic understanding of that, then we can really appreciate and understand the effects of neurostimulation. So I want to start by thanking the support for this work. So this work I'm going to present today was supported by 2 grants from NIH, National Institute of Aging, and NIMH, and also by the generous support of Boston Scientific that provided us the implantable pulse generators that I'm going to talk about in my talk in a little while. So as I mentioned, it is essential to understand the basic mechanisms, the neural mechanisms that mediate cognitive functions, and the one cognitive function that we have relied in particular is working memory. So bear with me then as I review just a general overview. What are the mechanisms that allow neurons in the brain to maintain information in working memory? So we can train animals to do working memory tasks. Here I'm going to show the simplest first order working memory task that we can train a monkey to do, or a rat or a mouse to do, which is a delayed response task. So we ask subjects to fixate on a screen. We showed a visual stimulus somewhere in the screen with a white square. The subject has to remember where in the screen that stimulus appeared over a period of a few seconds, which we call that the delay period. And then when the fixation point comes off, that's a cue for the subject to tell us where that stimulus appeared, and the subject communicates that to us by making an eye movement to wherever they think the stimulus appeared. So it's been known for half a century that when you perform a task like that, there are neurons in the prefrontal cortex and other areas of the brain that respond during the presentation of the visual stimulus here with an increased firing rate, but they also continue to discharge and continue to generate action potentials even during the delay interval when nothing is happening anymore, just a fixation point has come on. And furthermore, this activity in the delay period is selective for the location of the cue, where the cue is presented. So that provides a neural correlate then of spatial working memory. There's been some debate in the literature recently about whether this is the only mechanism or the predominant. There are other mechanisms, and I don't have time to go into much detail to that. But suffice to say, for this, at least for this type of working memory path that we're discussing, visual spatial working memory, there's very strong evidence that this persistent activity is what determines behavior and what subjects recall. So the first question here is, how do neurons generate persistent activity when nothing is happening? There's no stimulus anymore in the screen, and yet these neurons continue to discharge. Well, it turns out this is not a super difficult engineering problem. This is sort of a cartoon version of the simplest circuit that could generate persistent activity. And here, each triangle here is supposed to represent a single stimulus. A single neuron, which is excited by this afferent input. But then this neuron is connected with all the other neurons in the network. And even when this afferent input, the visual stimulus in this case, is no longer present, the neurons will continue to generate activity, and this will continue to reverberate in the network. So for this visual spatial working memory that we're discussing about, we can take a step further, basically, and we describe this network as a ring. So here in this ring, the position of each neuron is such as to represent the location of the stimulus that this neuron is most selective for, most sensitive for. So this would then be a neuron that is maximally activated when a stimulus appears at the top of the screen at the 90 degrees, basically, in this circle. And if we then design a circuit where the strength of connections between neurons depends on their spatial preference, neurons that have very similar spatial preference are strongly connected with each other. Neurons that have orthogonal preference are very weakly connected. Neurons that have opposite preference are not connected with each other. Just a very simple network like that can really capture the behavior of real neurons that I described. And I have a little animation here if I can turn that on. So this is then a very, exactly this network now represented in this fashion. I have taken this ring of neurons and spread it in the x-axis, and the y-axis in this plot is the firing rate of each neuron. So we see in real time what happens in the screen. When a stimulus appears at this location 0, the neurons whose preferred location is there are activated. Neurons that are nearby are activated to a less extent. But interestingly, when the stimulus is no longer present, this activity continues to be present, continues to survive purely by the virtue of connections between neurons that continue to discharge with each other. So this very simple network then captures what neurons are really doing in the prefrontal cortex. Okay. So this model also provides an explanation for the inaccuracies of memories then. If a true stimulus appears right here at the upper location, then neurons that are whose preferred location is at 9 degrees will be activated. But then this activity during the course of the delay period may drift just by random noise in the system, may drift basically randomly from this original location. And then what the subject will recall is where this peak of activity is at the time that the subject has to make an movement. And this model is called the bump attractor model, because this network of neurons behaves as an attractor, and then there is a bump or peak of activity in the network, basically, that determines what is the stimulus that we recall. So this is a model, but there's strong evidence, basically, that this is true in spatial working memory. And a very simple illustration of that is shown here. So suppose that in some trials, this bump of activity drifts to the left relative to the real location of the stimulus. Suppose in other trials, this bump of activity drifts to the right of the real location. If that were true, then the activity of neurons on trials when the subject recalls something leftwards relative to the real stimulus and rightwards relative to the real stimulus, they should have different levels of delayed period activity. And this is sort of an experiment that we did a few years back now, and in fact, we saw that this is the case. So if we look at the peak of activity of individual neurons in trials that we separated that have clockwise deviations and counterclockwise deviations from the real location of the stimulus, the tuning curves of these trials basically differ slightly but systematically. Okay, very good. So armed with this understanding of the basic mechanism of working memory generation and maintenance in the population of neurons in the prefrontal cortex, we can now see, can we improve working memory, and can we use neuromodulation to alter the activity of these neurons? And we're going to use this model then to interpret our results, because that's the main mechanism that allows the maintenance of working memory. All right, so the neuromodulation that we have looked into using is targeting the nucleus basalis of Maynard. The nucleus basalis is the exclusive source of acetylcholine in the neocortex in primates and in humans, therefore provides us with a target to essentially to put our finger on the switch of acetylcholine release in the neocortex, basically by means of electrical stimulation. The strategy of our experiment is shown here. So we implant one electrode targeting the nucleus basalis. So these are deep brain stimulation electrode. But at the same time, we can record neural activity. We can have a recording cylinder over the prefrontal cortex and record also activity and find out truly the effects of this neuromodulation on prefrontal neurons. These are histological slides that we got out of a post-mortem out of one of the first animals that we implanted, just to verify that the electrode is in fact where it's supposed to be, and these are neuron stains with chat in the vicinity of the electrode. So we have verified that this is in fact our target. Now, a critical, of course, parameter here, a critical question is the stimulation parameters, and this is with what we started, and I'm going to show you in a second how we had to deviate a bit from this plan. So we used a 200 microampere biphasic stimulation. The duration of a pulse is 100 microseconds, and we started with a frequency of 80 hertz of continuous stimulation. Modeling the effect of static stimulation using established models in the field suggests that we are inducing a radius of activity or a diameter of activation about 0.9 millimeters. So to show that in scale with a monkey brain, we're really exciting very small volume around the nucleus basalis. So that's the strength and the power of deep brain stimulation that allows us very specific targeting of the area that we want to stimulate. All right, so we started then this experiment. We implanted monkeys. We had them do a working memory task, and we applied 80 hertz continuous stimulation inspired by the movement disorder protocols of stimulation and Parkinson's disease stimulation. And what we saw is, much to our disappointment and chagrin, the 80 hertz continuous stimulation not only did it not improve performance of these monkeys doing the task, but in fact very dramatically, very systematically decreased performance. So if you apply 80 hertz stimulation of the nucleus basalis, we saw that the performance of the monkeys relative to the control condition systematically decreased. So, we then scratched our head basically and tried to sort of titrate and figure out maybe if we apply stimulation at different time intervals during the task, we're going to see a different effect. Let's see which part of the task is important. But as it turned out, it didn't even matter synchronizing the stimulation with a part of the task. When we started playing with limiting the stimulation in time and using an intermittent protocol of stimulation, so we used 20 seconds of stimulation and then just wait for 40 seconds, then we saw very consistently, and we've replicated this result now in multiple animals in 2 different laboratories, or 3 different laboratories in 3 different universities, and we now saw that this protocol of intermittent stimulation improves working memory performance. This is 2 different subjects. This is the control performance in their task. When we apply the stimulation now in this fashion, the monkeys do much better and consistently across the entire range of delay intervals. The stimulated animals with this protocol do better. So we played with the parameters a little bit, and this 60 hertz is not necessary. So somewhere in the order of 60 to 80 hertz, we have found that the magic number is about 1,200 pulses per minute. So we can apply 60 hertz for 20 seconds or 80 hertz for 15 seconds. But it is imperative that we do that continuously. And we don't know quite yet, what is the reason, why the continuous stimulation doesn't work. Sort of conceptualize that as some kind of a depletion of acetylcholine. If you exhaust the resources, you don't get quite an effect. This short burst of activity is what allows the system to continue to provide. But we have ongoing experiments to really find out exactly how is the release of acetylcholine regulated. All right, so having established this behavioral effect, we went on now to understand what is it that this protocol of stimulation, what does it do in neural activity? And we used a slight variation of this working memory task that I described to you. We now use two stimuli, not just one. We present one stimulus, the monkey has to wait over delay, and then a second stimulus. And depending on the color of the fixation point, the subject has to remember either the first stimulus and make an eye movement towards the location of the first stimulus and ignore the second stimulus, or if the fixation point is blue and the subject has to ignore the first stimulus and has to remember where the second stimulus appears and then make an eye movement towards the location of the second stimulus. So we call that the remember first or remember second task that we use just to probe this higher level of working memory, more and more complex task. The fact that we employ this intermittent stimulation protocol actually worked very well, integrated very well with our recordings. If the entire trial here, the fixation interval, stimulus presentation, delay, second stimulus presentation, second delay, if that entire trial lasts for 10 seconds, we can present 4 or 5 trials basically over a period of 1 minute and then have a 15-second intertrial interval between, longer intertrial interval between these trials, apply our stimulation here, and then continue. So our stimulation is always applied in the intertrial interval, not when the subject is doing a task at that exact moment, but we're able to squeeze this stimulation basically without interfering with the performance of the task. And of course, we have some sessions where the stimulation is not applied, so we always compare activity and performance in trials that are always structured in this fashion. So when we did this experiment, so the main effect that we saw is that the overall effect that we saw is that nucleus B-cell stimulation increases activity in the prefrontal cortex. So the blue line here represents firing rate of neurons in the control condition. The yellow line here represents activity in the stimulation condition. And we see that overall, the activity of this one neuron example, when we applied stimulation, was elevated. But quite surprisingly and quite unexpectedly for us, when we plotted the activity across all the neurons in the prefrontal cortex that were recorded during stimulation, we saw that, yes, this activity under stimulation in the yellow curve here is higher than the activity in the blue curve. However, what we have done here is that we have broadened the selectivity of neurons. And we see higher activity now, even for these locations that neurons were not very selective before. This was true both when we looked at the stimulation period and also for the delay period of the task. And in the single neuron sample, you can see it here as well. So we have this increase in activity even for the non-preferred location of the neuron. That's what we're seeing over here. So for me personally, it was very difficult result, to deal with. So as Ben mentioned, I did my postdoctoral training in the lab of Pat Coleman-Rakis, and one of your big discoveries has been that D1 antagonists stall working memory. So when you apply D1 antagonists to the prefrontal cortex, that creates tuning out of neurons, basically, and that since then, this development of tuning by any kind of drug is interpreted as a sign that this drug improves behavior. You have some neurons that become selected for some information, and the subject presumably becomes better at this task because the neurons signal better the information. But here we see the opposite. So we see neurons in some way lose their tuning, have more broad tuning under nucleus-based stimulation. So, to really understand what's happening here, so we employed the Baumgartner model, and these are work that we did in collaboration with Albert Comte, which is a computational neuroscientist who was one of the people who developed this model in the first place. And in fact, we see how this effect of neurostimulation can account for the changes in behavioral performance that we saw. And the main result is illustrated here. So we're seeing, again, one of these maps of neural activity across the network of the neurons. The y-axis, again, represents different neurons that have different preference for different kind of stimuli. The x-axis here is time. So on the left panel, we see what would happen when you present a stimulus at the 50-degree location here. The stimulus is no longer present. There's a delay. And then we have the second stimulus also that appears over here. So during the course of the trial, this bump of activity may drift. So the location that we recall at the end of this activity may slightly differ, and we have some slight error relative to the original location of the stimulus. So now on the right, we see how we would expect the network to behave when we have this broader peak of activity under cholinergic neuromodulation. And what we saw very consistently in this stimulation is that when you have these broader peaks, we tend to have less drift. The peak or the bump of activity will have more inertia, if you will, and you have less error then by virtue of this bigger and broader peak, basically, that doesn't shift very much in time, and it's less resistant to distracting stimuli. The model, however, makes a very interesting prediction. So in general, that would be the case. This broadening of receptive fields for this kind of task is going to be beneficial. However, there are some special cases where this is detrimental. And such a special case is shown over here. So if after the first stimulus presentation, we have a second stimulus that is close enough to this bump of activity, then the two bumps may merge with each other, and the second stimulus may, in fact, pull away from the original location, and the subject is going to be confused when the second stimulus appears, and therefore the activity is going to drift. So that sort of explains and reconciles our findings that when you have broader kind of peaks and the neurons are less selected, you do lose something. You lose this fine-grained stimulation, this fine-grained ability to remember details, very precise details of stimuli. However, for the most part, the effects of stimulation are beneficial. And this prediction of the model was born by the experimental results. So this is the model predicted performance for stimuli that are far away from each other or close to each other. And we in fact show that the stimulation in general improves performance, it improves fidelity of memory. However, we lose some precision for these very special cases. Another funny prediction of the model is that when you bring the excitation of the network at this higher level through neurostimulation, and you have some expectation for stimuli, this network with the stimulation on sometimes creates bumps of activity even when no stimulus is presented. So we call those phantom bumps of activity. And in fact, we saw that in the experimental results. The blue line here is responses of a neuron in the control condition. That red line is responses of the same neuron during stimulation. In a CATS trial, when we didn't show a stimulus, and in fact, the monkey was expecting for one exactly at this time point, but the stimulus never appeared. So we saw prefrontal neurons fire when nothing was actually present in the screen. So these results all say that neuromodulation by means of stimulating the cholinergic forebrain can improve performance. We have some very clear understanding of how that happens mechanistically, and there are some special cases that this may be detrimental, but overall, this increased level of activity is going to improve cognitive function, is going to improve working memory. I should say these are all results that we obtained in young adult monkeys that presumably had plenty of acetylcholine endogenously generated in the prefrontal cortex, and we were still able to improve their performance above those levels. But the next thing that we wanted to raise is what happens in aged monkeys that may in fact have more need of cholinergic stimulation, and can we then improve working memory in these animals, and can we use this knowledge then to translate into human conditions. And we're in the middle of doing these experiments now. These are unpublished results that I'm going to show you. So in order to prepare the ground for a potential clinical trial, we implanted monkeys with the human devices. So those are FDA-approved stimulators from Boston Scientific. And these are implanted subcutaneously, and the electrodes are the deep brain stimulation electrodes target the nucleus basalis, but then these are wireless devices that we can control remotely. The monkeys perform that task in their cages, so they have a touch panel that they operate, and we use a working memory task, a delayed match to sample task, where the monkey has to remember a stimulus, and then after a delayed period, we show two different choices. We can do these experiments on an adaptive manner every day. So we start with very short delay intervals, and we increase. So every day, basically for different conditions, we can find out the threshold. What is the longest delay that the monkey consistently perform these tasks? And these are the results then from the first two animals that completed our protocol of 8 weeks of stimulation. The y-axis here is performance. The y-axis is the longest delay interval that the monkeys can do reliably. This is the baseline condition, and this is the condition under intermittent stimulation of the nucleus basalis. And as I say, this baseline was established over a period of years. So these animals have been doing this task for a very long time. And then after the stimulation, we saw a quite dramatic increase in performance. And I should say, it didn't matter, again, if the stimulation was synchronous at the time that the monkey was performing this task, or if this stimulation was done at a different time of day. Over the 8 period of weeks, applying this stimulation to the nucleus basalis improved the ability of these animals to do this task and improve their working memory performance. And I should say, this is a protocol of 1 hour of stimulation every day over 8 weeks. These are the same results here, shown in more detail. These are 2 subjects, baseline and unilateral stimulation. And for the second subject, we have also moved from unilateral to bilateral stimulation. And bilateral stimulation so far appears to contribute a further benefit than unilateral stimulation of the nucleus basalis. These are the curves of performance over the entire delay interval. And the reason that we did unilateral, we started with unilateral stimulation, is that we wanted to have another measure of nucleus basalis activation into brain activity, which we obtained through PET. And these are, again, our 2 subjects. So we use the unstimulated side as a control. Both of those animals are stimulated on the right side, and you can see higher metabolic activity of the right side in both of those animals after 8 weeks of stimulation. And this is PET imaging that we obtain at rest when the animal is anesthetized. So the stimulation has produced some enduring change in metabolic rate that it's visible on the stimulated side. So this is where we stand now. To summarize our results, the bump attractor model of prefrontal cortical persistent activity predicts working memory performance. By using cholinergic deep brain stimulation, targeting the nucleus basalis of Maynard, we can improve working memory, and we do that by virtue of increased prefrontal cortical persistent activity, which stabilizes the attractor and, except for some special cases, will generally make working memory better. And we now have shown that performance of aged animals improves with nucleus basalis stimulation, and it also increases PET metabolic activity of aged monkeys. I'm going to stop here and thank the people who actually did this work. These are my lab members highlighted. Those are everybody who participated in this project. And I also want to thank our collaborators in this project, Albert Comte, who did the computational work, David Blake, who did some of the initial experiments, the behavioral experiments with deep brain stimulation, Dario Engelot and Sarah Big, who are neurosurgeons at BUMC, who have been part of this project from the beginning since we started at Vanderbilt. And we're now we're laying the groundwork for some human trials in Alzheimer's patients using the same approach. And finally, Cholinergic Chief, who did some of the neurophysiological experiments at Wake Forest. Thank you very much.
All right, we're going to take some questions from the audience. Just raise your hand if you have a question. While we're waiting, okay, here, let's start here.
I hope this is on. Okay. My question has to do with how subject-specific this response is. Did you do any, maybe you mentioned it and I missed it, did you do any extinction studies within subject, dose them down with a blocker to see what the effect of the stimulation was?
So this is, these are very good questions. We've done a variety of experiments and we have more that we want to do. The furthest we have done is by using simultaneous stimulation and drugs that the monkey. So if we were to give nucleus basalis stimulation and donepezil at the same time, we saw no further improvement. So either of these interventions, donepezil itself or nucleus basalis stimulation itself, improved performance, but both they reached some kind of ceiling. And we also were able to diminish this effect with muscarinic blockers, so cholinergic blockers as well. So, ultimately, we want to get true measurements of acetylcholine. It's a little more difficult to do that. In the monkey, we have parallel rodent experiments that do that. But by all accounts, by indirect evidence at least, we are increasing release of acetylcholine. That's relevant for the effect.
I wonder if you could talk a little bit about the relationship between working memory, the classic Goldman-Rakish working memory paradigm, and Alzheimer's. Are those, how much of a leap is it to go from one to the other?
Yeah, so that's another very good question. So we use working memory as a proxy of general cognitive ability. So Alzheimer's disease, obviously the biggest and more obvious impediment that patients have is the loss of long-term memory, or anterograde amnesia. That's the kind of thing that strikes the most. But we feel that improving working memory, we're just going to improve the general cognitive function across a domain, basically, of function. It may not necessarily stop this progression that leads to degeneration, but it is a more effective treatment than we have available now relative to cholinergic drugs, for example.
Do you see other changes in these monkeys after the stimulation? Nothing obvious. We've trained them to do a narrow range of tasks. These monkeys look normal. They look perfectly fine. And the effect of stimulation does not seem to attenuate with time. So these effects seem to be enduring, lasting. So we're trying to see here for how long this is effective. So that's a question that is going to be also important for translate that. All right, great. Let's see. We have a question back here.
Thank you for the presentation. Question. So it sounds like you have kind of cumulative effects over the time when the memory is proven over the time. Do you think it is of interest to explore if you can scale down to even less than an hour per day stimulation as you improve your memory over time, you kind of achieve certain performance, and now maybe you don't need as much to continue going?
That's also a very good question. The parameter space is obviously vast. I don't know if it's infinite, but it's huge. The question here, the core of the question is that by applying the stimulation, have we changed something permanently, and can we take it off at some point, or is it necessary to apply that continuously to see an effect? And it seems that we do need it. It seems that we have, so far, preliminary results suggest that if we stop the stimulation, then these animals' performance is slowly going to decline again. So it seems that this continuous stimulation is essential for maintaining this benefit.
All right, let's do one last question.
It's a question from the chat room. Can you compare the stimulation protocol to that of patients used in human DBS?
Yes. So the protocol of stimulation of human DBS for movement disorders is essentially what we started at the beginning. And we know that this does not work. And I think the reason for that is quite clear now, that DBS protocol for movement disorders stimulates neurons at so high level that they cannot keep up. I think we saw some results of that yesterday, that eventually the end result of this stimulation is inhibitory. It inhibits the output of the basal ganglia. So here, we don't have an overactive cholinergic system. Quite the opposite. We have hypoactive cholinergic system. So we need some protocol that will increase cholinergic release, and will do so in a continuous fashion, in a prolonged fashion, without depleting that and without exhausting it. So that's my understanding for that question.
All right, thank you very much. One more round of applause. Our second talk in this session is going to be by Diane Rizzuto, who's coming from Nia Therapeutics, kind of a representative of the idea that you can do really, really great and really interesting science outside of the halls of academia. Of course, Dan has a really impressive academic resume. He did his PhD at Brandeis with this kind of world-leading learning and memory group there, centered on John Listman. And then he moved to Caltech and did some really exciting work there, and got tangled up with Mike Kahana, and they started this company. He's the leader of the company. He's going to talk about some of the work that they're doing and some of the really exciting directions that they're going.
Thank you very much. I'm excited to be here. Thanks to the organizers for inviting me, and I'm looking forward to sharing our latest research on how to improve human verbal memory using closed-loop neurostimulation, and how we're applying this to memory loss in traumatic brain injury. So first up, my disclosures. I own more than 5% equity in Nia Therapeutics, which is developing a closed-loop device for treating memory loss. Learning goals for this talk. Memory loss represents a large societal unmet need. I probably don't need to say that today. It's pretty obvious. And neuromodulation is really an emerging clinical treatment for memory loss. A key part of our strategy relies on the fact that patterns of electrical activity across the brain predict verbal memory performance. And this is a critical fact that we really base our therapy on. And the effect of stimulation depends critically on these brain states that we've identified, these whole brain activity. And we've developed, accordingly, a closed-loop neuromodulation therapy based on stimulating lateral temporal cortex, which has been shown to reliably improve verbal memory performance. So first, Memory loss is a huge unmet need. 27 million Americans, 1 in 12 people, currently suffer from some form of memory loss due to a number of underlying conditions. From traumatic brain injury, most people don't know, but memory loss is actually the number one symptom of a moderate to severe traumatic brain injury. It can also be caused by mild cognitive impairment, which is kind of a halfway point between age-related memory loss and Alzheimer's disease. It's sometimes seen as an early form of Alzheimer's disease. Then there's Alzheimer's disease itself, which is a huge global burden on its own. And finally, age-related memory loss. This is normally seen and observed across the lifespan, that memory performance declines in some people more than others. And what are the symptoms of memory loss? They can be subtle at first, but it often involves forgetting appointments and events. These patients have trouble with finances, cooking, driving, finding their way around town. They often withdraw from social activities because of embarrassment about their condition, and they ultimately end up unemployed as a result of the impairments in cognition, which greatly disrupts quality of life. And it's devastating not only to patients, but to their families as well. And currently, no effective treatments are available. Yeah, there's a few drugs available for Alzheimer's disease, but if we're being honest with ourselves, they don't actually work. And so new approaches are needed, new modalities of treatment, new theories for the underlying disorders. And clinicians are increasingly using neurostimulation to treat a variety of neurological disorders. Parkinson's, epilepsy, blindness, deafness, all of these things have been approved by the FDA over the past 20 years as indications for neurostimulation. And the global neurostim market is close to $6 billion and growing very quickly. So we're really riding a huge market adoption wave today in neuromodulation. And researchers have used electrocorticographic direct recordings of the human brain to study human memory for decades. Epilepsy patients, we've heard across many different talks today, are used as a prep, as it were, for studying various neurological disorders. And these patients are often implanted for 1 to 2 weeks with cortical and subcortical leads, and for the purposes of diagnosing the foci of their epilepsy for potential curative surgical treatment. And during the time that they're in the hospital, researchers administer tests, collect ECOG, electrocorticographic activity, to uncover the neural basis of memory. It's actually pretty exciting. And our team measures verbal memory function using the free recall task. Verbal memory is the form of memory most impacted in patients with memory loss. It's used to diagnose the transition from healthy aging to Alzheimer's disease. It's used to diagnose mild cognitive impairment. And to measure verbal memory, you typically present a word list. Oftentimes there's some distractor period to distract them from rehearsing the word list, and then the patient's instructed to retrieve as many words as they can from that word list. In our case, we present 12 words, one at a time, on a computer monitor. They then do some math problems to distract them from rehearsing the list, and then they retrieve as many words as they can in a 30-second period. A whole list takes about 1.5 to 2 minutes to conduct. And you have to repeat that because memory performance is so variable. You need to repeat that process many times. In our case, we repeat it. 25 different lists are presented in a session to get an accurate assessment and measurement of that patient's memory performance at that point in time. And so what we found, a kind of a foundational approach, and I want to make sure everybody understands, this key graph, because the rest of the talk really relies on this, is that human verbal memory performance is predicted by changes in spectral power across the brain. And so I'm presenting here data from 98 patients that were collected as part of our studies during the free recall task. And what I'm doing is I'm comparing spectral power at frequencies from 2 hertz all the way up to 95 hertz on the x-axis here. And I'm comparing it for words at encoding while words are presented, but we're sorting words and comparing them for words that are later recalled versus words that are forgotten. So there's an inherent control condition in the free recall task. Some words are remembered and some aren't. And so we compare at encoding when you don't necessarily know which words are going to be recalled or not. And this is what you find. There's an increase in high-frequency activity that predicts or is correlated with recall of items later, and a decrease in low-frequency activity. And you see this across the brain. I'll show brain montages in a minute. But that's the general pattern of effect. An increase of high-frequency activity and a decrease of low-frequency activity predicts later recall. And using this signature, we've identified a memory network across temporal and frontal lobes. I'm showing a video here across 100 some odd patients showing the pattern of high-frequency activity as it evolves as a word is presented, the word on. You see it coming from occipital lobe to temporal lobe, then to frontal lobe as the word gets turned off, and then the pattern repeats as the next word is presented. And based on this deep understanding of the electrophysiological basis of memory, DARPA funded our team in 2014 to develop a closed-loop neurostimulation therapy for memory. And this was called the Restoring Active Memory Project. It was a $24 million contract to the University of Pennsylvania. And we enrolled over 400 epilepsy patients across 8 clinical centers around the country. We prototyped a 256-channel closed-loop neurostimulation system with our partners at Medtronic, and we administered direct brain stimulation during memory performance to evaluate different strategies for improving memory or modulating memory. And this project produced one of the world's largest brain sensing and stimulation data sets. As I mentioned, behavioral data from over 400 patients performing memory tasks with and without stimulation. Electrocorticographic data from over 30,000 macro electrodes. And we've released these data publicly. You can go to this link to get a partial data release from the project. As I mentioned, we built a portable 256-channel neuromodulation system for use in the epilepsy neuromonitoring unit. It had 256 channels for sensing, 128 channels for stimulation, collected data at 1,000 hertz suitable for LFPs or ECOG activity, and it was able to communicate with a laptop via Wi-Fi or USB. And you can see here the setup. Patients were implanted with percutaneous leads during their stay in the epilepsy monitoring unit. We used the ENS, as we called it, to collect brain activity and stimulate the brain. This was controlled by a host laptop that had the control interface for the ENS, and then a task laptop presented these behavioral tasks to the patients. And we trained machine learning classifiers to predict memory performance in real time. We used spectral decomposition, so we transformed each ECOG channel into its spectral components, power at different frequency bands. We sorted words at encoding again, right? We used the same effect I showed earlier into words that were later recalled or forgotten, and we trained a logistic regression classifier to identify the biomarkers that predicted ultimate recall or forgetting. And we could use these to predict in real time a patient's performance on the task. What I'm showing you here is the classifier output across 8 different lists for one of our patients. And when the classifier output is above 0.5, that means the classifier had strong confidence that that word would be remembered. And when the classifier output was below 0.5, that means that the classifier was confident that that word would be forgotten. And as you can see, the ultimate recalled or forgotten words are indicated in red or blue here. Most of the patient's recalled words were classified above 0.5, meaning that the classifier was fairly accurate at classifying those. And accordingly, most of the forgotten words had a classification of below 0.5. And we used a k-fold cross-validation technique to prevent overfitting of these data. And across the cohort of 102 patients that participated in this study, we had a mean area under the curve of 0.63, which may not mean a lot to everybody, but AUC is the way that you evaluate a machine learning classifier's ability to classify training sets, and this is highly significant. So we then built or evaluated different neurostimulation strategies to evaluate the impact on memory function. And interestingly, the effect of stimulation depends critically on the state of ongoing oscillations across the brain. So again, we sorted memory encoding events into poor memory states and good memory states according to those electrophysiological signatures, that increase in high-frequency activity that I mentioned earlier. And some of the lists included stimulation, some of the lists did not. This was a randomized, blinded, and sham-controlled study design. Patients didn't know which lists included stim, which ones didn't. They were blind to that randomization, and it was sham-controlled, where each patient acted as their own control. And so 0 in this graph represents a patient's baseline of memory performance, and they either were increased with stim or decreased with stim. And what we see was that the brain state matters. At the time that stimulation arrived, if they were in a poor memory state, as indicated by the low graph on the left, you actually saw an increase in memory function. The recall performance was greater when stimulation arrived at a poor memory state. Conversely, if they were already in a good memory state as assessed by this whole brain classification, then stimulation actually impaired memory on average. And so this told us that a closed-loop approach to using neurostimulation is likely to be required. You don't want to stimulate when the brain's in a good memory state. You want to wait until it's in this poor state that we've identified. And so we then did the closed-loop study in a new cohort of about 25 patients. These patients were stimulated only during poor memory states, as indicated by our real-time classification schema that I presented earlier. Again, a randomized, controlled, and blinded, sham-controlled study design. And what we found was that before we had honed in on the timing of stimulation, needing to be during these poor memory states, now we're honing in on the target of stimulation. And what we found was that closed-loop stimulation improved memory only when it was delivered to lateral temporal cortex. And that's the blue bar on the left. Conversely, when any other area, or when summing across all the other areas we stimulated, even closed-loop neurostimulation impaired memory. So we've now demonstrated the timing and the target that are necessary in order to make a reliable improvement in memory function. And now we're focusing in on the indication that we find to be the most interesting right now, which is memory loss following traumatic brain injury. It turns out that traumatic brain injury is a risk factor for developing epilepsy later in life. So just by chance, many of the patients that we studied had a history of moderate to severe traumatic brain injury. This is a new cohort of 8 patients with a history of moderate to severe TBI. We administered closed-loop neurostimulation of lateral temporal cortex only during poor memory states in a randomized, blinded, sham-controlled study design, and we found a 19% improvement in list learning across the cohort. And that's shown here on the right in those gray bars. We've ranked them according to efficacy, and with the average being 19 in that red bar on the right. And you'll probably notice that the effect was highly variable across these patients. And we believe that we can further optimize therapy performance by optimizing electrode placement. Because these patients were in the hospital for epilepsy, not for memory, the surgical plan and the electrodes were placed for purposes of epilepsy, not for memory. And you can see here on the right that there is a large variability in the placement of the lateral temporal cortex stimulating lead. And we're now honing in on the hotspot within lateral temporal cortex that drives this effect. And so we believe we can further improve efficacy in our next clinical trial. So I think, a movie is worth 1,000 figures. So I'm going to show you one of the RAM technology in action. This is one of our patients at Mayo Clinic who participated in the study. You'll see this is one list that he participated in. You'll see 12 words being presented. He'll do some math problems and then try and recall as many of the words as he can in this from the preceding list. Yellow words indicate words that are accompanied by stimulation. So he's viewing these on the Mac laptop that's in front of him. There will be a test. Now he's doing some math problems to distract him from that list. 20 seconds of basic arithmetic. So he just recalled all 12 items from that 12-item list, and I challenge any of you to match that in your daily life. And that was his comment afterward when the experimenter was incredulous that he had just done this. And he said, I had a good picture in my head. I could just see. So something about this list helped him visualize these words much better than he had been before. And that was, his subjective experience. And we've published on this particular patient, and we stimulated lateral temporal cortex, and we got the effect. We stimulated medial temporal cortex, and we did not get the effect. Back to lateral, got the effect. So we've done washouts and shown that it really comes down to stimulating this precise target. And so we've now demonstrated improvements in list learning with closed-loop neuromodulation of lateral temporal cortex. So what? That's sometimes the question that I get. So you've shown increases in laboratory memory tests. Well, it turns out that verbal memory performance, these word list learning, is associated with reduced disability and increased productivity across many different patient indications. As I mentioned before, the loss of verbal memory and in the free recall task is used to diagnose Alzheimer's disease. So it is the metric by which we gauge the degree of impairment. In patients with multiple sclerosis who go through these outbreaks of MS, the degree to which they lose verbal memory and list learning directly predicts their transition from employed to unemployed. It's one of the few things that does predict that. And in patients with traumatic brain injury who often recover spontaneously after their TBI, the recovery of verbal memory after the injury is the only thing that predicts their ability to return to work. And if they're lucky enough to return to work, it's the only thing that predicts their on-the-job performance. And it also predicts long-term reductions in disability. So this is a critical outcome measure that we believe will drive functional improvements in patient populations. So based on these demonstrations, Nia Therapeutics is now developing the smart neurostimulation system. And memory loss due to traumatic brain injury is our first clinical target. We believe that the smart neurostimulation system is personalized neuromodulation for memory disorders. Here's a concept of the Nia system. It includes 4 depth leads with 16 contacts each for interfacing with the brain. There's a cranial implant with 64 sensing channels and 2 stimulation channels. There's an earpiece that provides power to the implant and provides circuitry for closed-loop data processing. It does the analysis and controls therapy delivery. And it has secure cloud connectivity to a cloud-based AI platform that personalizes the therapy to each patient. And we believe this is the next generation system for closed-loop neuromodulation. We've prototyped our commercial platform. I'm showing you the current prototypes of the earpiece, the implant, and our cloud-based AI. And we're advancing towards preclinical safety testing later this year to demonstrate safety before we go to IDE-approved studies by the FDA. And our platform can generate massive data sets to increase the pace of therapy innovation. It has USB for high-speed connectivity in the clinic, streaming 64 channels of 1,000 hertz data, Bluetooth for low-bandwidth data collection at home, and easy integration of behavioral and neural data sets to identify new biomarkers, refine existing biomarkers that will lead to therapy improvements over time. And I think I'm speaking to the choir here when I say that new indications for closed-loop neuromodulation are on the horizon. People are exploring Alzheimer's disease, mild cognitive impairment, anxiety, depression, OCD. The NIH has estimated that over 100 million Americans suffer from nervous system disorders with a huge cost to society, and neuromodulation can be part of the solution. So I hope that I've convinced you that neuromodulation can be part of the solution for patients with memory loss. It critically depends on the state of the brain, and so having a sensing and stimulating and closed-loop approach is critical. Thank you very much.
I want to make it clear, Nia is not a sponsor of the conference, so there's no conflict here.
I wish we had enough money to do that.
We'll be bumming you up.
Hey, thanks for the great talk. The different cognitive strategies that participants use might be able to change their memory performance quite dramatically on a list test like the one you showed. How did you control for that sort of variability in subjects?
It's a great point. And what you find is that performance generally solidifies. So patients there's the initial lists, we throw out the first few lists that each patient performs in a given session as they're warming up, getting reacclimated to the task, and potentially using a different strategy, as you're pointing out. But patients typically default to, once they've practiced the list a few times, to a consistent strategy. There are strategies that are better than others. Visualizing the word list and combining words into a single monolithic visualization can often yield the best results in depths of encoding. But what you don't find is over time that patients are really switching their strategies. They tend to fall into a single one.
Okay, thanks.
Hi, over here. I'm Karen Maxon, University of California, Davis. I was at Thomas Jefferson University when you guys first started this many years ago, and it's very exciting to see how far it's come along. It was a really great talk.
Thank you.
I want to go back to your part about when to stimulate, and you talked a little bit about identifying the state, and if the state was a good memory state and you stimulate, you have poor response. Can you just define the state a little bit better and talk about how easy it is to define that state and what constitutes it? Thanks.
And This is the foundational result. It's called the subsequent memory effect. And I would refer you to Burke et al. 2014, who've really illustrated this. And what you're doing is you're comparing, during the encoding of words, the spectral power for words that are later recalled versus words that are forgotten. So that's the internal control of this task, is some words are remembered and some words are forgotten. And some of that can be due to exogenous factors, but most of that, as has been published by my collaborator Mike Kahana subsequently, most of that is due to endogenous brain activity, endogenous changes in brain function drive which words will later be recalled and which words will be forgotten. And this is the biomarker, it's called the subsequent memory effect, that determines which words will be recalled and which words will be forgotten. So this is how we gauge, this is how our machine learning classifiers are trained on these features: an increase in high-frequency activity above 30 hertz and a decrease in low-frequency activity below 30 hertz. That tilt in the power spectrum predicts memory activity.
Okay, I have a couple questions. In TBI and also in deep brain stimulation, which you have a combination of both, there is a saying, there is no such thing as a true sham in human models, right? So in that context, the subjects that you were implanting, what was the age of acquisition of injury for them, and how long after acquisition of that injury were you able to go through these tests with them? I mean, what sort of plasticity may have occurred in between? And also the age of onset of injury is very important. So that's one question. And then I'll ask another one later.
Yeah, so these patients typically had their injury many years prior. This was not a recent injury. They typically received an injury in early adulthood or late adolescence, and then developed epilepsy later on in life. And then even later on, they were evaluated for surgical resection to cure their epilepsy. So many years, typically 5+ years, occurred between the TBI, the index injury, and our recordings. And I would oftentimes true sham is not possible in DBS because everybody knows they have a DBS system. But what we've shown here is acute changes, and we can interleave stim and no stim in a true blinded sham control. So we believe we do have a really solid sham control.
Okay. Then my other question has to do with, what we typically call the jeopardy effect. You know you're going to be tested. You prime yourself for it, right? No matter how many tests you're given, you are really on the spot, ready to do the job. How does that state of mind relate to a typical everyday life scenario where you might be walking down the road and try to memorize your grocery list, right?
Yeah.
How do you get rid of the jeopardy effect?
That is the million-dollar question. And essentially what we've shown today is that we can see acute changes in memory function on these list learning tasks. And the million-dollar question is how these acute changes that we see transform are reflected in activities of daily living as somebody's going about their life, shopping for groceries, etc. And we do believe that there's strong linkage in the existing literature because performance on these tasks does correlate, but that is the million-dollar question, that we have to show that. We have to show not only that we see these changes, and so in our design with the FDA, we've agreed that the primary outcome measure will be CVLT, which is this California Verbal Learning Task, list learning task, and the secondary outcome measures are more functional measures that will measure the activities of daily living in each patient. And we're going to have to show both in order to get FDA approval and reimbursement for this therapy.
All right, we'll take one last question.
Hi, thanks for the cool presentation. Can you clarify what stimulation parameters you use when you're applying the closed-loop therapy?
Yeah.
And what is kind of your theory behind the physiology of how this is changing the brain state when you apply stimulation, especially in the context of like how you showed this memory state is a whole brain network kind of thing going on?
Yeah, so another great question, and I'll see if I can, in terms of the electrode, I didn't show the slide, sorry. In terms of the parameters, we're doing high frequency stimulation, either 100 hertz or 200 hertz frequency, 300 microsecond pulse width, amplitudes around 1 milliamp. That's all published in our ESIOT 2018 manuscript, so I encourage you to review that. In terms of the mechanism of action, we're actually showing when you stimulate at the right time and at the right place, you see broad changes reflected in that biomarker that I showed you. You see a poor memory state get transformed into a good memory state. The classifier output increases from below 0.5 to above 0.5 with stimulation. So and that comes down to again, the timing and the placement of stimulation being able to affect through functional connectivity, broad scale brain activity. Thank you.
Right. Thank you very much. Let's do one more round of applause for our speaker. All right. Great. Well, we're going to have one more talk in this session on learning and memory. Dr. Diane Chan is coming from MIT. She did her undergraduate at Cornell University, then did an MD, PhD at Boston University, became a neurologist. And she did a lot of kind of low-level research on mice and then got very interested in applying this to humans. That's what brings her to the research that she's doing right now in Liwei Tsai's lab at MIT and the work that we're going to hear about today. So I'm looking forward to this too.
Thank you so much to the organizers for inviting us all for this very exciting symposium, and of course for the opportunity for me to share this work with you. So I'm here to talk to you a little bit about some non-invasive neurostimulation, which is a little different from all the topics thus far. And as we know, Alzheimer's disease is a big problem these days. Over 47 million people in the world suffer from Alzheimer's. Patients with Alzheimer's come in with cognitive impairment, but by the time they see me in the clinic, they already have the classic pathological findings that we see on autopsy, which is buildup of amyloid plaques, neurofibrillary tau tangles, neuronal death causing structural abnormalities, including atrophy of the brain, synaptic loss, and neuroinflammation. In such a multifaceted disease, it's been really hard to develop a therapeutic that's disease-modifying. And so that's why we're looking into how neuronal networks or a systemic kind of therapy would be effective in Alzheimer's. So in Alzheimer's disease, we're really interested in gamma oscillations, which is a type of brainwave that is important for higher cognitive functions, including working memory, sensory processing, and spatial orientation. And so gamma frequency oscillations are between 30 and 80 hertz. And as you can see in this cartoon, it is propagated across all these neurons here. What's interesting for Alzheimer's is that in patients with Alzheimer's, you can see that there is desynchronization of the gamma frequency oscillations between different parts of the brain. In mouse models, we can also see that there's reduced 40 hertz signal, reduced amplitude in the gamma frequency range, reduced network stability, resulting in reduced cognitive function. And this is all due to the PIV interneurons that are dysfunctional, causing this kind of destabilization. In the Scilab, we're very interested to know how early gamma waves are disrupted. Here are some histology results looking at staining for amyloid. So early on in this 5xFAD mouse model, you can see that there's some deposits of amyloid here that is brown. It's not until the mice are 6 months old that we start to see some memory impairments in their performance of behavioral tasks. But we can actually see disruptions in gamma oscillations as early as 3 months in this mouse model. So what we did was we recorded a mouse who's running through a virtual maze, running over this ball here. We're recording their LFPs, and we're finding that in awake behaving mice who are 3 months old 5X FAD mice, there's reductions in gamma oscillations. So let me just show you the data here. In wild-type mice, you can see that there's a good amount of gamma power during restful periods. But in the Alzheimer's disease model, there's reductions in gamma frequency oscillations. So what we think is that actually early in the disease course, perhaps even in people, there's disruptions in gamma oscillations. We don't know if this is because of the pathology of Alzheimer's disease itself, or does it precede the deposition of amyloid, tau, and the development of cognitive impairment? The Scilab, in collaboration with Emory Brown and Edward Boyden at MIT, hypothesized that perhaps by boosting gamma power, we would be able to reverse some of these pathological findings that we see in Alzheimer's disease. And initially, these experiments were done using optogenetics, where electrodes were implanted into the hippocampus, and mice were stimulated using optogenetics to increase 40 Hz oscillations. Surprisingly, these mice had reductions in amyloid and tau deposits in the brain. And so the next step was to see if we could do this non-invasively, because clearly we cannot do optogenetics in people. And so using our 5 senses, we've decided to try and see if we could entrain the brain using light and sound. The first experiments are done with light alone, and you can see here there's a mouse in a cage, and you can't see the flickering, but the lights, the LED lights here are flickering at 40 Hertz, controlled by our Arduino system. And in recording the paraspectral density, you can see that 40 Hertz light flicker increases gamma oscillations or increases 40 Hertz entrainment in the brain. And this recording is done in the visual cortex. And even after just 1 hour of using this light flicker at 40 Hertz, we could see that there is reductions in amyloid, that this is the soluble amyloid using ELISA. As compared to control conditions, you can see there's 50% reduction in soluble amyloid in the brain here. The other control conditions that we used in mouse models include just bright light. We used 80 Hertz light and sound. We used other frequencies, including 20 Hertz. And so this seemed pretty specific to 40 Hertz. And what I'm not showing here is all the hard work that was done in finding 40 Hertz as the right frequency, at least for mice. We've used 35, 40, 45, 50, 55, and so on and so forth. And so we did find that 40 Hertz was the best, and we added a synchronized sound to this light stimuli. And we found that after prolonged treatment, meaning 1 hour a day for over 6 weeks, we could see that there's reductions in deposits of amyloid. Now, this is Clarity Brain from the 5X FAD mice, and here is a mouse staining for amyloid with no stimulation. There's deposits of amyloid throughout the brain. But using combined light and sound at 40 Hertz, you can see, if this video will play, that there is actually reductions in amyloid throughout the brain, not just in the visual or auditory cortexes. Now, what about the other pathological markers of Alzheimer's disease? So we decided that we would look at phosphorylated tau, which is in neurofibrillary tau tangles, in a P301S mouse model. So with stimulation, you can see this is what a classic neuron looks like with neurofibrillary tangles, staining for phospho-tau. But with gamma stimulation, this is light at 40 hertz, stimulating the mice for an hour daily for 3 weeks. You can see that there's reductions in phosphorylated tau as well. And so this is just a summary, but looking at different areas of the brain, not just in the visual cortex, but also in the somatic sensory cortex, the hippocampus, and the cingulate cortex. There is reductions in amyloid staining after using 40 hertz light. There's reductions in amyloid as well as in tau. The initial areas where 40 hertz entrainment occurs using light and sound, of course, are in the auditory and visual cortexes. But with repeated stimuli, an hour a day, every day for 6 weeks, let's say, you can see that there are areas of entrainment, in the mouse model at least, throughout the brain. In addition, you can see with our clarity brain earlier that reductions in Alzheimer's-related pathologies can affect the entire brain if you use the stimuli for longer than just a day. In this different, more severe mouse model of Alzheimer's disease, the CKP25 mouse model, we can see that by turning on CKP25, there is reductions in brain volume altogether. This is a good model for Alzheimer's because that's what happens in a human as well. There's neuronal loss and there is atrophy of the brain. You can also see that the ventricles are dilated here in this mouse model as a result of this atrophy. But using 40 hertz light in this case, on a daily basis for 6 weeks, we can actually prevent this brain atrophy from occurring in this mouse model. The question is, how is this happening? We still don't know the answer to that, unfortunately, but the lab is working very hard on that at MIT. And so some of the things that we saw that was very robust, and we didn't know why, was that the microglia were very much activated, both in increasing its numbers of cells of microglia, but also in the morphology of the cell. We saw that the microglia actually it had increased amyloid deposits in the bellies of these cells. So the question is whether microglia are activated by this 40 hertz light and sound stimulation, and they're going around and cleaning up the amyloid, or if there's also increased excretion of these waste products, because we also see that there's vasodilation that occurs, first in the visual cortex, and with repeated stimulation, you can see this vasodilation in other parts of the brain as well. But most importantly, and most exciting for us, is that these mice actually did much better in their behavioral testing. So some of the tests that we did were novel object recognition, novel object location, and like I said, gamma frequency oscillations are important for sensory processing and spatial orientation. So those are the tests that we chose. And also there's a MACE test. But they did better on these behavioral testing. And so, with all these exciting preclinical data, we decided to see if we can translate this work to be done in people. This is just a summary of all the findings that we described just earlier, that using GENIS, or the gamma entrainment using sensory stimulation, there's reductions in amyloid, in phospho-tau, activated microglia and increases in vasodilation, and these mice also had improvements in their behavioral testing. So this is the exciting part. Where I came in back in 2018, we developed several prototypes of devices to use light and sound stimulation, or GENIS, in humans. And I'll show you some data from that. But we used EEG to record the brain's response. So in 2018, we did our phase 1 study where we optimized for safety and feasibility in healthy young and older volunteers and in patients with mild Alzheimer's disease. And then we did a small pilot phase 2 study where we sent these devices home with people who had mild Alzheimer's dementia and asked them to use our device for an hour a day. It's very important to us that people are very compliant with usage at home. As you know, you can turn on the device and leave. And so we really wanted everybody to be able to turn on the device in the first place because these are patients with mild dementia and that they were able to use it every day. That is safe to use. And we did some basic outcome measure testing. And we are launching a larger randomized control trial now. So in our phase 1 study, these are the 3 cohorts of patients and participants. I'll show you some data from that. The important piece of that was that there were very few reported adverse events. None of them were severe. And so people reported that they were a little bit drowsy, a little bit sleepy, and they might feel that they had some dry eyes. But nobody had any seizures, luckily. And the EEG was reviewed by myself and as well as an epileptologist who's not related to the study. So this is the EEG data that we got from our combined light and sound stimulation in the cognitively normal group who are younger, older, as well as people who had mild Alzheimer's disease. We saw that using our light and sound, this one is the light and sound together, you can see that there is increases at 40 hertz here. The blue line is the baseline recording, so our device is on, but the light is occluded, just so that we know that we're not recording any kind of noise from the device itself. The light and sound together really induced the strongest entrainment. As you can see here, this is the light alone, the topography map. The sound alone, and then the combined light and sound. You can see where on the brain there's 40 hertz activity, and you can see that the combined light and sound actually induced the most cortical engagement. Now, we do ask our participants to use our device for an hour a day every day. And we didn't know if people would adapt to this kind of visual and auditory stimulation. So we did the EEG recording for a whole hour. And here the dotted line is when we turn on the device, and the other dotted line is when we turn off the device. And you can see that we didn't draw this line in. This is actually the ERSP here. This is the 40 hertz power that you can see across throughout the whole hour. So happily, no one adapted to the stimulation itself, and they were in training the entire time. We also were interested to see if there were changes in connectivity, which we heard a lot about yesterday. Using EEG and phase lag index, we looked at some connectivity data from our EEG, and we saw that using the combined light and sound, of course, induced the most connectivity, better in the young groups than in the older or the Alzheimer's groups, but that's known in the literature. And so we chose to use the light and sound for our clinical trials because it induced the strongest 40 hertz entrainment in terms of power, but also increased connectivity as well. Now, we were fortunate to collaborate with people who are doing studies in epilepsy. So in patients who had medically intractable epilepsy scheduled for intracranial recordings, we were able to show them our light and sound device and record their EEG. And we found that not only do we have induced entrainment on cortical structures as measured by our surface EEG, we also see entrainment in deeper subcortical structures that are important for Alzheimer's disease. So in the gyrus rectus, this is the 40 hertz peak here, the gyrus rectus, the amygdala, the anterior hippocampus, and the posterior insula. And this is the blue line is, of course, the baseline recording where the device is on but the light is occluded. So for our phase 2 study, it's a pilot study where we had our patients take our devices home to use the device every day for an hour a day. And then we asked as a primary outcome measures whether the device was used appropriately every day and if it was safe to do so. And then we had some exploratory outcome measures, and so I'll show you some of the data from that. We only had 15 people randomized 1-to-1 to the control settings versus the active settings of 40 hertz light and sound. And so this is the device that we sent home with them. You'll notice that there is a iPad here. So the thing we learned from phase 1 was that if people were not paying attention or if they were asleep during stimulation, there was not as much entrainment. And so we actually put an iPad here so that people's attention was focused to the center of the device and we're playing whatever fun movies they are interested in watching, and compliance and engagement was much higher. And so we were excited to see that there were no severe adverse effects of using our device at home every day for an hour a day for here 3 months. And this is how we track compliance. So our device has a counter on it that tells you if it's on or off, but easily somebody could turn it on and leave or go and make a sandwich for themselves. And so we use some machine learning to do some eye tracking to make sure that, there's a video being recorded of everybody using the device at home. But now we know the subject is present, if their head is drooping and nodding off, or if they're looking towards our device, or if their eyes are open or closed. So hopefully everybody is awake. And so with this composite score, we were able to measure compliance, and we found that actually a lot of people were able to be very compliant with usage, up to 96% of the time. But one participant was not as compliant, as you can see here, 61%. And so that's good in that you don't really see a split between the control group and the active group. We're worried that somebody might figure out they're in the control group and they'll stop using it. But it seemed like everybody was as compliant as each other. Using structural MRI brains from these people, we were able to measure brain volume, just like in our mouse model, CKP25 mice. You can see the healthy brain looks like this, coronals. And in mild Alzheimer's disease, you can see global atrophy on the outside of the brain and also ventricular dilation. And here we could see that using our light and sound Janus at home every day for this is a 3-month time point, there is preservation, I guess, of the brain structure in that the control group actually had increased brain atrophy. This is expected in the natural progression of Alzheimer's disease, but in the active group we were able to perhaps prevent that. There's also hippocampal atrophy that happens in Alzheimer's. That's in the natural progression of Alzheimer's, which you can see in this control group, and it's significant from baseline, but our active group did not show significant atrophy over the same time frame. This is a very small cohort of patients, and so although these are encouraging results, you have to take it with a grain of salt. We used functional MRI to look at the default mode network, and we found that the active group getting 40 Hz light and sound Janus had increased connectivity with the posterior cingulate cortex as compared to the control group who had reductions with connectivity in that area. And we also looked at the medial visual network, where we found that there were increases in connectivity in the active group, while the control group stayed about the same. We sent actigraphy devices home with these patients as well to look at sleep. because poor sleep can exacerbate pathology in Alzheimer's disease and mediate cognitive decline. And we found that there was actually improvement in interdaily stability, which is a measure of circadian rhythmicity in these patients with Alzheimer's who used our active devices. Meanwhile, the control group had reductions in their circadian rhythmicity. And then the last thing we looked at, is a face name association test, which is a sensitive test of memory in this mild dementia group, where this is difficult to do in a conference like this size, where you have to learn people's names. And so we had the patients try and learn everybody's name here, and then the test was to see if the patient knew that this person was Wanda and if this person was Benjamin. And so we saw that the patients who had our 40 Hz Genus device had improvements in their accuracy in learning people's names, whereas the control group stayed about the same from baseline, maybe a little bit worse. We hypothesized that maybe because of our visual stimulation, we were able to kind of improve connectivity with the medial visual network, as we can see here, and that these improvements in this kind of visually based cognitive test was related to this increase in connectivity there. And so in summary, we found that using Genus was safe in human participants, and that we were able to effectively entrain the brain of both healthy, cognitively normal people as well as patients with mild Alzheimer's disease. We were happy to show that there was target engagement of the brain, not only on cortical areas measured by the surface EEG, but also in subcortical areas that are important in Alzheimer's disease. We found that perhaps using Genus, we were able to prevent atrophy of the brain over this short time frame and improve sleep and also associative memory. Our group is not the only group doing these studies. Nia Therapeutics also did a study on 76 patients with mild Alzheimer's, treating them an hour a day every day for 6 months. And they found that there was actually less decline in these activities of daily living in this cohort who used the Genus device. They had slower worsening of their memory and cognitive scores using MMSE. And again, they saw that there was less atrophy of the brain over this time frame. There's a small cohort of patients with MCI who also used this device, a different device, and an Emory, who found that it was safe and tolerable to use, and that there were changes in connectivity as well. And so, thank you everybody for the opportunity for us to present this work. We're very grateful for our patients who have used this device every day, like for now actually 2.5 years at home. An hour a day. And so we're very grateful to them for all their hard work. And of course, I need to thank my mentors, Li Wei Sai, who spearheaded this work with Ed Boyden and Emory Brown at MIT, and the whole Genus team. So thank you for your attention.
That was absolutely fascinating. Phenomenal work. I have a question. So my grandmother affectionately called most electronic devices idiot boxes. And pretty much every single piece of electronics that anybody's ever used, right, probably has a built-in flicker rate of 60 hertz.
Yeah.
Are these idiot boxes, are these things making us sick in some way?
Well, luckily, these 60 hertz idiot boxes are flickering, not really flickering, because their duty cycle is 100%. So your brain isn't really in training, per se, to 60 hertz. And we just heard that 60 hertz might be actually beneficial in certain parts of the brain. But you're right, we don't really know the effects of the electronics and the media that we face today. There are frequencies we found in mouse models of Alzheimer's that are detrimental, that might increase amyloid deposits in the brain, at least in mouse models. And so you're right, we don't know whether this is detrimental to us using all these devices. But I think that because the duty cycle is 100% here, I think we're safe for now.
Hi, thank you for the work you're doing. So my question is, since there's no adverse effects from 1 hour a day while they're like fully attentive to it, is there any evidence to suggest that if it was on all day, 24/7, even if they weren't like fully attentive to it, that there still would be no adverse effects?
That's a great question. And wouldn't we love to develop a immersive experience with using Genus? But I think we've learned a lot from Dan's talk, actually, that there are good brain states and bad brain states, where stimulation during certain times might be beneficial or detrimental to memory. The problem with doing this all the time is that we don't even really know the effects of this 1 hour stimulation yet. But there have been groups who have asked patients to do the stimulation for 2 hours a day or 4 hours a day. But again, they have to drop everything and do it for that time frame. And so, compliance is the issue, I think, for that. But once we do figure out the science behind this, it wouldn't be terrible to make it an immersive experience, in that it's built into all our media already.
Hi, I had a quick question. Thank you. Super interesting. I was curious if patients themselves noticed a difference as well?
Yes. And so if they are sitting and looking at both devices at the same time, you will notice the difference between the two.
Do I have to stand to make it work, really? Okay.
No?
Sorry, I did not phrase that well. What I meant was, did they notice a memory improvement themselves, just in their everyday lives?
Well, there's a lot of anecdotal reports. They feel clearer in their memory, that they feel like, their spouses think that they're doing better over this time frame. But we did have longer term data, which I did not show, that does show stabilization of memory over 2.5 years of usage. And so it's not a significant improvement from their baseline. But at least over 2.5 years, you would expect these patients with mild Alzheimer's dementia to decline in their memory. They just seem longer. The other thing is that we are supposedly, here improving sleep. And so the improvement in sleep can make you feel clearer, as we all know this morning with coffee and whatnot. That having that coffee and having a good night's sleep would help you feel better, that you can access your memory in a clearer way. And so we still don't know if this device is improving the sleep and therefore improving memory, or if it actually is improving pathology and doing the work that way.
Sorry.
My question's related to that. If sleep is being improved, I may have missed it, but what did you say, the control stimulus? Was there stimulus in the control patients? Because I could imagine, if these patients are getting an hour of extra light a day, that might be resetting the circadian rhythm itself, and then you're seeing all these great effects?
Yeah, I did not describe it in detail, but the control setting is constant light and white noise with the same amount of light and sound. So 400 lux of sound on both groups and 68 decibels of sound on both groups. Both groups are getting light therapy. It's not the amount that I would recommend to my patients for sleep improvement. Like I tell patients to get 10,000 lux at a tangential angle for your face for like 30 minutes, an hour a day in the morning. And so it's a dimmer light than that, of course. But they're both groups are getting the same amount of light and for the same amount of time.
Let's thank Dr. Chan one more time. Thank you.